Welcome to the Narako Deep Dive, the show where we break down the latest research and science around learning, memory, and on some days, anything else that catches our eyes. So let's jump right in. Today's topic. I hope you enjoy. Doctors have spent decades blaming one villain for erasing memories in Alzheimer's. Sticky little protein clumps called amyloids.
The bad guy. Textbook.
Right. Except here's the part that breaks my brain. Without those exact same clumps, you couldn't remember your own name. Not yesterday's name. Not your name. Right now.
Which sounds completely backwards.
It's like finding out the termites eating your house are also the carpenters holding it up. This is the Narako Deep Dive. Today, why your brain deliberately builds the same clumps behind Alzheimer's. To make memories permanent.
On purpose.
Try this. Think of a smell from 20 years ago. Your grandma's kitchen. A specific perfume. Whatever it is for you. It's just there. Instantly. Like it never left.
Mm-hmm.
Now here's the thing that wrecked me when I first read this. The actual physical stuff storing that memory, the proteins in your brain cells, none of them are 20 years old.
Nope.
Like, not even 20 months old. Your body is tearing down and rebuilding most of those proteins every few days.
Every few days, yeah. It's constant turnover. Break it down, recycle the parts, build it back up.
Okay, wait. That can't be right. If the actual hardware is dissolving and reforming on a loop, how does the memory not just dissolve with it?
Right. This is the paradox.
It's like your hard drive is melting down and reforming every 3 days, but somehow the same file keeps opening.
That's exactly the puzzle biologists have been sitting with. The experience is permanent. The parts are not.
So something has to be keeping the shape of the memory, even while the pieces underneath it get swapped out.
Yes, and for decades, nobody could point to what that something was.
But now they can.
Now they can. And the answer comes down to a single protein, one that behaves completely differently depending on its shape.
One protein doing this whole magic trick.
In fruit flies, where most of this work was done, it's called ORB2.
ORB2.
And in us, in mammals, there's an equivalent version called CPEB.
CPEB. Okay, so somewhere in there is the actual mechanism that keeps a 20-year-old smell locked in place.
That's exactly where we're headed.
Because I need to know how a protein that gets destroyed every few days somehow doesn't lose the plot.
So picture the synapse. That's just the tiny gap where one neuron hands a signal to the next one, like a docking bay. Most of the time, ORB2 is just hanging out there, alone, a single floating molecule, what biologists call a monomer. And in this form, it's not building anything. It's actually doing the opposite.
Wait, the opposite? So it's blocking things?
Exactly. It's a repressor. And here's the part that gets me. It goes after mRNA, which is basically the genetic blueprint the cell reads when it wants to manufacture a protein. ORB2, in its lonely monomer form, seeks that blueprint out and physically clings to it.
Okay, so it's like sitting on the instructions.
Kind of, but it's more specific than just sitting on it. It targets one particular part of the mRNA called the poly-A tail. Think of that tail as a protective cap on the end of the blueprint. As long as it's long enough, the cell's machinery can grab it and start reading. Actually, you know what? It's almost like a fuse on a stick of dynamite.
Okay, I like that. Go on.
The singulars, Right, so as long as that fuse is a certain length, the whole thing is primed and ready to go off, meaning ready to get built into protein. But ORB2, in its monomer form, actively chews that tail down shorter, and a shortened fuse just doesn't light. The blueprint's still there, technically intact, but nothing can read it anymore.
So it's hidden in plain sight.
Hidden in plain sight. Yeah, that's a great way to put it. And this matters enormously because without that mechanism, your synapses would just be manufacturing new structural proteins constantly, randomly, for absolutely no reason.
Which sounds exhausting, honestly.
It would be. Biologically speaking, it'd be chaos. You do not want a neuron building permanent new hardware every single time a random photon hits your retina or a random gust of wind touches your skin. So the resting state, the default, quiet, nothing's happening state, that's orb-2 doing its job as a roadblock. It's basically the system holding its breath.
Right. So most of the time your brain is just on standby.
Exactly. Standby. Waiting. And the whole system stays like that. Fuse cut short, blueprint locked away, nothing being built, until something actually happens out in the world that the brain decides is worth remembering.
Okay, so this whole time we've been talking about the brakes, which means somewhere there's a gas pedal.
There absolutely is. And that's where it gets wild.
Okay, I need this next part immediately.
Picture the synapse actually getting hit with something worth remembering. Not wind on your skin, but real signal. A jolt, a taste, a face that matters. And when that happens, orb-2 does something no one expected for decades. Instead of staying a lone floating molecule, it starts sticking to other copies of itself. It aggregates. It physically clumps into that same kind of protein tangle we associate with Alzheimer's, an amyloid.
Mm-hmm.
And here's the part that gets me every time I explain it. The clumping doesn't break the protein, it flips it. As a lone monomer, remember, orb-2 was the roadblock, shortening that fuse on the mRNA blueprint, keeping things shut down. The second it locks into that amyloid shape, its job reverses completely. Now it protects the fuse instead of cutting it. It holds the blueprint open.
Wait, the exact same molecule just folded differently does the opposite job.
The opposite job. Total reversal, purely from shape. And once that blueprint's exposed, you get this localized burst. New proteins get built right there, right at that one synapse, in that one spot the brain just decided mattered. Those fresh proteins physically thicken and reinforce the connection. That's not a metaphor for a stronger memory. That is a stronger memory, structurally, in real tissue.
Okay, that's... wow.
It gets better. This amyloid is prion-like, which just means it's self-copying. It doesn't need a fresh signal to keep existing. It grabs newly made orb-2 as it floats by and folds it into that exact same clumped shape, over and over. Researchers call it a conformational memory stored in a shape, not in any one physical molecule.
Oh. Oh, wow. So that's the answer to the paradox from before. The proteins keep degrading and getting replaced every few days. But the shape just keeps rebuilding itself?
Exactly that. The parts are disposable. The shape isn't.
The hard drive keeps melting and reforming, but the file survives because the shape of the file keeps stamping itself onto whatever new material shows up.
That's the cleanest way anyone's put it to me.
Okay, but now I need to know why this clump is the hero of the story and the Alzheimer's version is the villain. Because on paper, you just described a protein tangle building up in a brain cell, and that sentence usually ends badly. Because here's my problem with it. If I ran a factory, and one day all my machinery just clumped together into one giant tangled pile on the floor, that's not precision manufacturing. That's a disaster. That's the day the plant shuts down. So how does a tangled protein clump start doing delicate, precise work instead of just jamming everything up? And honestly, I want to push further than that. Are we even sure this is real? Like, maybe some scientist took a bunch of orb-2 protein, stressed it out in a dish somehow, It clumped up because stressed proteins do that, and everyone's reading way too much meaning into what's basically an accident of the lab bench.
That skepticism is exactly what the researchers had to sit with, too. It's not a dumb question. It's actually the central methodological problem of this whole field.
Okay, so walk me through it. How do you rule that out?
Well, the easy path would have been to just grow the protein synthetically, what's called a recombinant protein, meaning you insert the gene into bacteria and let the bacteria manufacture it for you in a vat. Cheap, fast, you get tons of it.
Sounds efficient.
It is, and it's also not good enough evidence here, because bacteria aren't neurons. A real brain cell applies all these subtle chemical finishing touches to a protein as it's built, little tags and modifications, and bacteria just don't do that. So lab-grown ORB2 doesn't necessarily fold or behave the way it would inside an actual synapse.
So you could clump bacteria-grown ORB2 all day, and it wouldn't prove anything about what's actually happening in a fly's head.
Right. It would prove protein chemistry can produce a clump under stress, which nobody doubted. It wouldn't prove the brain does this on purpose as a functional, controlled thing.
Which means if you actually want to settle this, no shortcuts. You need the real protein, made by a real neuron, sitting exactly where it naturally sits.
Exactly. What researchers call the endogenous protein, naturally occurring straight from a living fly brain, no substitutes.
Okay, but a synapse is microscopic. How do you even get enough of that to study?
That is a genuinely wild story.
So there's this researcher, Ruben Ervas, and his job was basically to go find this naturally occurring ORB2 sitting inside real fly synapses, not grown in a bacterial vat, pulled straight out of a living brain, exactly where it forms.
Right, the endogenous protein.
Endogenous, yeah. But here's the problem. A synapse is unbelievably tiny, and the amount of this protein sitting in one fly brain is just nothing, practically undetectable. So to get enough of it to actually study, he had to scale this thing up to a number that I read in the source material, and genuinely had to stop and reread.
Okay, how bad is it?
He harvested ORB2 from roughly 3 million adult fruit fly heads.
3 million.
3 million individual fly heads, dissected essentially, to isolate this one protein in quantities you could actually work with.
The sheer manual labor behind that number is staggering. That's not a weekend in the lab. That's years of somebody's life.
Right. Somewhere there is a very tired grad student. But that scale was the whole point. You need that much material before you can actually see the structure.
And to see it, they didn't use anything like a regular microscope.
No. This is cryo-electron microscopy, cryo-EM. You flash freeze the purified protein instantly. So fast, the water around it can't even form ice crystals. And then you fire electrons at it and use the pattern to reconstruct a 3D map.
It's like taking thousands of snapshots from every possible angle and letting a computer build the object back up in 3 dimensions.
Exactly. And the resolution they got down to was 2.6 angstroms.
Which doesn't mean much until you translate it.
Right. An angstrom is roughly the width of a single atom. So this isn't a blurry blob under a microscope. This is an atomic-level blueprint. They weren't just confirming a clump exists. They were mapping, atom by atom, the physical shape of a memory.
Which is exactly what they needed, because that structure is about to tell us the whole story of why this one is safe. What they found were filaments, long protein fibers, about 75 nanometers, so incredibly thin, twisted into this elegant 3-fold symmetric shape, almost like a braid with 3 strands wound around each other. But the part that mattered most wasn't the outside shape at all. It was buried at the very center of the fiber.
The core. Okay, why does the core matter more than the shape?
Because the core is what decides whether the protein behaves or goes rogue. In Alzheimer's, the toxic amyloids have what's called a hydrophobic core, meaning the center of the clump literally hates water. And since a cell is basically a bag of water, a water-fearing core is a huge problem. Those proteins panic. They fold inward to hide that core from the water around them. And in doing that, they clump onto anything nearby, completely indiscriminately.
Which is what tears through the neuron.
Exactly. It punches holes in cell membranes. It's chaotic. It's unregulated. That's the villain version. But when Hervas mapped ORB2, the memory-forming amyloid, the core was the opposite, hydrophilic. It actually attracts water, and it's held together by these tightly interlocking glutamine residues. Think of them as little chemical hooks that lock into each other with real precision, not panic.
Okay. So instead of a protein desperately hiding from the water,
it's completely at home in it, stable, calm, structurally happy, just floating there.
Okay, this is where I want the Lego version. Because in my head, the toxic one is like, you panic, you super glue a Lego set together to keep it from falling apart, and now it's ruined forever. It's rigid, it's a mess, nothing can ever click into it again.
That's a great image, and honestly, not far off. That's the hydrophobic disaster in a nutshell.
So then ORB2 is, what, the good Lego clasp?
To build on that, yes. It's like one of those specialized Lego pieces, engineered with an actual clasp mechanism. It locks in tight, it holds real structural weight, but it was designed to click that way. It's intentional. Nothing about it is an accident of chemistry.
Which, hang on, that's the whole point, isn't it? The hydrophilic core is basically proof this thing was built on purpose.
That's exactly the significance. It's not biological trash accumulating with age. It's an architectural choice the cell evolved, stable enough to hold a memory for decades, but built in a way the cell can still work with, rather than something that just wrecks everything around it.
Okay, so the same basic move, folding into a clump, becomes either a wrecking ball or a load-bearing wall, depending on one tiny detail buried at the center.
One tiny detail. That's the whole difference between disease and memory.
Okay, wait. Before we go any further, I need to talk about the name of this chaperone protein, because it might be my favorite detail in this whole story.
Here we go.
So technically, in the paper, it's called CG10375, which sounds like a parking validation code, not the molecule that builds permanent memory.
Mm-hmm. Very romantic.
Right? It's got what's called a J domain, basically a specific molecular shape that lets it dock onto other proteins, like a plug finding the right socket. But nobody's putting J domain on a t-shirt.
No, they are not.
So the researchers gave it an actual nickname. They called it Funes, after the Borges short story, Funes the Memorious, about a man cursed to remember literally everything. Every leaf, every cloud, every second of his life. Forever.
Which, once you know what this protein does, is an unbelievably good name.
It's so good. Somewhere there's a scientist staring at cryo-EM data at 2 a.m. going, this needs a literary reference.
Scientists love a dramatic name way more than people give us credit for.
Secretly, you're all frustrated novelists.
Guilty. But Funes earns it, because this chaperone does the exact opposite of what a chaperone is supposed to do.
Wait, opposite how?
So think about what chaperone proteins are supposed to do in literally every other context in biology. They're the cell's janitorial staff, quality control. Their whole job historically is to patrol around and make sure proteins fold into the correct shape. And if something folds wrong, they either force it back into shape or they tag it for the cell's garbage disposal system so it gets broken down before it can cause trouble.
Right, the tidy-up crew.
Exactly. That's the job description for basically every chaperone we've studied for decades. Prevent misfolding or clean up misfolding. Funes reads that job description and does the opposite. It doesn't stop the fold. It causes it.
Hold on. It's actively pushing the protein toward the wrong shape?
Toward the amyloid shape, yes, which every other chaperone in the cell would treat as an emergency. And here's how it physically does it. ORB2, remember, starts as a lone monomer, a single floating molecule. When a synapse gets strongly stimulated, some of those monomers start sticking together, but not instantly into the full, locked amyloid. First, they pass through this in-between stage called an oligomer, which is just a small, loose huddle, maybe a handful of ORB2 molecules stuck together, unstable, not yet committed to a shape.
Mm-hmm.
And that oligomer stage is actually the dangerous moment. In a lot of disease biology, that's exactly the intermediate step that goes rogue and starts recruiting everything nearby into a toxic mess. Funes physically grabs the ORB2 protein right there in that loose, unstable huddle. And instead of breaking it apart, it forces the transition forward. It locks the oligomer into the final, structurally stable amyloid, the one with that hydrophilic core we talked about, the one that actually turns on protein synthesis at the synapse.
So it's not preventing the misfold. It's finishing it.
It's finishing it, on purpose, at exactly the right moment. And that's the part that gets me every time I think about it, because it means the field's entire 100-year definition of what a chaperone does was incomplete. We thought chaperones only existed to stop bad things from happening to proteins.
And Funes is out here building something.
It's a construction foreman, not a janitor. It shows up on site specifically to make sure the structure gets built correctly and gets locked in, which honestly reframes chaperones as, functionally, architects of memory. Not just cleanup crews standing around waiting for a mistake.
Okay, that's genuinely a little mind-bending. So somewhere in your brain right now, there's a protein whose entire job is to force other proteins into a shape everyone assumed was always a disaster.
Every time you form a real memory, yeah. And that raises the obvious next question.
Which is?
If Funes is out there, grabbing ORB2 and forcing it into permanent shapes, what stops it from doing that constantly? Because you do not want a chaperone that's trigger-happy about cementing memories.
Right. You'd remember every grain of sand you ever stepped on.
Exactly the nightmare scenario. So there has to be a gatekeeper deciding when Funes even gets access to the material.
Okay, before we get to the gatekeeper, I want proof. Show me this actually happens in a living brain, not just a cryo-EM picture.
Fair demand. So the lab ran the classic fly memory test.
The smell and sugar one?
Right. Hungry fruit fly, one specific odor, immediately followed by a sugar reward.
Basic conditioning. Smell means food.
Exactly. Normally that memory fades after a while. But they didn't just watch it happen. They dialed Funes up and down.
Okay, round one, crank it up.
Long-term memory shoots through the roof.
Even with lazy, rushed training?
Even then, 1 week exposure, the kind that normally doesn't stick, and it sticks anyway.
So extra Funes basically force welds the memory in.
Pretty much.
All right, round two. Flip it.
They mutate Funes so it physically can't dock onto ORB2 anymore.
Can't grab it at all.
Can't grab it, can't fold it, nothing happens.
And the memory?
Gone. Long-term memory formation just fails.
Even with good, solid training this time?
Doesn't matter how clean the training is. No docking, no memory.
So it's not a dimmer switch. It's an on-off switch.
More Funes. Memory locks in harder. No Funes. Nothing locks in at all.
That's a genuinely clean experiment.
About as clean as behavioral neuroscience gets.
So chaperones aren't background janitors anymore.
No, they're the translator. The thing that turns one fleeting whiff of sugar into a permanent physical shape in the brain.
The difference between that happened and that happened to me forever.
Exactly that.
Okay, but that raises the obvious problem for me. If Foos is just standing there ready to grab ORB2 and lock it into permanent memory, What stops it from doing that constantly? Every random smell? Every boring Tuesday? Why isn't your brain permanently amyloiding everything?
That's exactly the right question. And the answer is honestly the most elegant part of the whole system. It turns out ORB2 isn't one thing. The same gene produces two different versions of the protein. Think of them as two flavors off the same recipe. One is called ORB2B, and it's everywhere. It's sitting at almost every synapse in your brain in huge quantities, quietly doing that baseline repressor job we talked about, keeping things shut, keeping the synapse resting.
Okay, the workhorse.
Right, but the other flavor, ORB2A, is the opposite. It's incredibly rare, and the cell keeps it that way on purpose through a chemical process called phosphorylation, basically tagging a protein with a little marker. You can think of that tag as a do not delete sign. Here's the twist, though. In a normal resting synapse, there's an enzyme called PP2A whose whole job is to rip that tag off.
Wait, so it deletes the delete protection.
Exactly. And without the tag, ORB2A is unstable. The cell's cleanup machinery recognizes it as unprotected and destroys it almost the instant it's made. So picture this constant, expensive little loop. Build ORB2A, strip its tag, shred it, over and over, just to guarantee there's never enough of it hanging around to accidentally spark an amyloid.
That's a lot of energy, just to prevent you from preventing a leaf.
It is, but that's the safeguard. Your brain would rather burn the fuel than risk cementing every random grain of sand into your synaptic architecture forever. Now, something meaningful actually happens. Real pain, or something intensely sweet, a genuine jolt. That triggers a flood of calcium into the synapse. And that calcium activates a completely different complex called TAUB/K. And TAUB-K overpowers the shredding. It slaps that protective phosphate tag onto ORB2A faster than PP2A can strip it off. For the first time, ORB2A survives.
It doesn't get deleted.
It doesn't. And that surviving ORB2A becomes the seed, the template. It's rare enough and specific enough that when it's finally stable, it pulls all that abundant waiting ORB2B toward it, locking it into that self-sustaining amyloid shape we mapped earlier.
So the rare one dictates a shape, and the common one just falls in line.
That's it exactly. Scarcity is the safety mechanism. The system only ever ignites when something is loud enough, biologically, to survive its own execution order.
Okay, that's genuinely beautiful. Nature built a lock that only opens for things that matter. Can I just try to hold this whole thing in one picture? Because I feel like if I don't do it right now, it's going to slip out of my head.
Please, go for it.
Okay, picture a house. And in every single room, there's a pile of dry firewood. Just stacked up, waiting. That's ORB2B. It's everywhere, it's abundant, and it does absolutely nothing until something tells it to. Now, somewhere in that house, there's a match. That's ORB2A. But here's the catch. The second that match sparks, someone runs in and blows it out. Every single time. That's PP2A.
The shredder.
The shredder, right. So for that match to actually stay lit, you need something strong enough to keep the flame alive even while it's actively being snuffed out. That's TAUB-K, the real event. The shock, the taste of something sweet, whatever's loud enough biologically to hold a flame against the wind.
That's a really good way to put it.
And once that match survives, it doesn't just set the wood pile on fire by itself. Someone still has to physically walk it over and touch it to the wood. That's Funes.
The chaperone.
The chaperone. It takes that one surviving spark and drives it straight into all that waiting ORB2B. And that's the moment the whole thing goes up. Permanent blaze. That's the memory.
I genuinely don't think I can improve on that one.
I'll take it.
But here's the part that actually gets me, building on what you just laid out. Every one of those safeguards, the constant extinguishing, the rarity of the match, the chaperone waiting there to strike it. None of that is waste. It's a filter.
A filter for what's actually worth keeping.
Right. The fire only ever catches when something out in the world is significant enough to survive all that resistance on the way in. Your brain isn't cheap with permanence. It makes an experience earn it. And that filter idea, by the way, isn't just a fruit fly thing. We've got the human version of this whole system. It's called CPEB3, and everything points to it running on the exact same amyloid logic.
Okay, hold on. I want to push on that for a second, because that's actually a massive leap, right? We watched a fly remember that a smell means sugar, and now we're talking about human psychiatric disease. That's a pretty big gap to just wave across.
It's a fair thing to be suspicious of.
Because I've seen this before. Someone finds a cool mechanism in an insect, and suddenly it's explaining depression, anxiety, whatever's trendy that week. So what's actually connecting the dots here? Is there real human data? Or is this just a nice story?
No, this is where it stops being speculation for me, honestly. Researchers ran genome-wide association studies, basically scanning enormous databases of human DNA looking for patterns. And they went looking at the genes that manage this exact chaperone system, the human cousins of Funes.
Mm-hmm.
And what came back wasn't noise. Variations in those specific genes are heavily linked to schizophrenia.
Wait, schizophrenia specifically? Not just generic memory problems?
Schizophrenia specifically. And once you sit with that, it kind of reframes the whole disease. Think about the core symptoms: hallucinations, a break from consensus reality, sensory input getting interpreted in a way that doesn't match what's actually out there.
Right. Hearing something that isn't happening, or being certain of something that just isn't true.
Exactly. Now put that next to what we just spent 20 minutes establishing, that chaperones like Funes are the literal architects that decide how a piece of sensory reality gets converted into permanent brain structure. So ask yourself, what happens when the architect misreads the blueprint?
Oh, so it's not that the person's imagining things on purpose, or their mind is somehow broken in this abstract way. It's that the actual construction crew at the synapse is building the wrong thing.
That's the suggestion, yeah. A malfunctioning chaperone could produce something that looks exactly like a reality distortion disease, but the root of it is purely physical, a miswired molecular machine, not a mysterious failure of the mind.
Which is a genuinely uncomfortable reframe. We tend to file schizophrenia under psychological, software, not hardware.
And this data is pushing back on that split pretty hard. It's suggesting some of what we call a software problem might actually be a hardware problem, sitting right there in one protein's shape.
Okay, that lands differently than I expected it to.
Which brings us right back to where this whole episode started: Alzheimer's. For about 30 years now, the entire strategy for treating it has been what I'd call blunt force. You spot amyloid plaques on a brain scan, and the goal, the entire pipeline of drugs built around this disease, is to dissolve them, break them up, clear them out wholesale wherever they show up, with no distinction made.
Because for decades, amyloid was just the villain. Full stop.
Right, and under the old model, that made total sense. If misfolded protein clumps are nothing but biological trash, then obviously you clean house. But we've just spent this whole episode establishing that some amyloids aren't trash at all. They're intentional. Built with a completely different internal architecture, remember? That hydrophilic core that actually likes water instead of panicking and tearing into membranes. And they're assembled under incredibly tight chaperone control, with Funes managing exactly when and where it's allowed to happen.
So if a drug just walks in and dissolves every amyloid clump it finds, indiscriminately,
it might not just be clearing toxic garbage. It could be tearing down structures the brain built on purpose. Memories, quite literally. which, incidentally, might be part of why some of these anti-amyloid drugs show cognitive side effects in trials that nobody's fully explained.
Wait, that's a pretty big deal, if that's actually true.
It's big enough that it's already reshaping how some labs approach the disease. The question isn't, how do we destroy amyloid anymore? It's, why did the management system fail? Remember, in a healthy synapse, there's this whole choreography running. ORB2A getting shredded unless a real stimulus stabilizes it. Funes only stepping in at exactly the right moment. Alzheimer's might not be amyloid appearing out of nowhere. It might be that regulation breaking down, the cleanup crew losing precision, so the clumps keep forming, or keep growing, long after there's any memory left to protect.
So instead of building a bigger sledgehammer,
We go find the broken quality control department instead. Study the actual chaperones, the proteins whose job is to take a functional amyloid apart once it's served its purpose, and figure out precisely where that machinery breaks down in disease. That's a completely different drug target than anything the field's been chasing for 30 years.
that's a wild pivot. We spend the whole episode arguing amyloid is secretly good, and now the fix is fixing the plumbing, not draining the pool.
That's not a bad way to put it, actually.
Terrifying and weirdly hopeful at the same time. Okay, but follow the logic all the way through for a second. If your brain has this whole detailed blueprint for building one of these memory clumps, the match, the firewood, the chaperone that strikes it, doesn't that basically guarantee there's a blueprint for taking it apart too? You don't design a lock without also designing a key.
That's exactly the inference the researchers make. Disassembly is probably just as regulated a process as assembly was.
Which matters way beyond Alzheimer's, right? Think about something like chronic trauma, PTSD, where one memory just won't let go. It keeps firing the same fear response years later, like the brain never got the memo that the danger's over.
Right, and under this model, that's not a metaphor. That memory could genuinely be sitting there as a hyperstable amyloid at the synapse. A structure so locked in, the normal disassembly instructions never get read.
So it's stuck on. The match is still lit, basically forever.
Exactly.
Which makes me wonder, what if you could just go tell it to stop? Not with a lobotomy, not with a sedative that numbs your whole brain to get at one memory. Just chemically nudge the local chaperones, the actual proteins that are supposed to unfold that structure once it's done its job, and let them do what they were built to do.
Unstrike the match.
Unstrike the match. You're not erasing a person. You're not touching the neuron itself. You'd be reaching in and dissolving the literal, physical, atomic thing that's holding on to the pain, the actual structure. And once it's gone, the synapse just exhales, goes back to resting state, like it was never lit at all.
That's the thought that stays with me from all of this research, honestly.
Yeah. The same molecule we spent this whole episode defending as the hero of memory might also be the thing that finally lets someone put down a memory they never asked to keep.
Which is a genuinely strange kind of hope to land on.
Strange, but I'll take it. All right, let's pull this all the way back out because we went deep into some fly brains today. 3 things I'm walking away with. 1. The reason your memories outlast the actual molecules in your brain is that it's never the molecules doing the remembering. It's the shape. The amyloid keeps rebuilding itself in the same fold, generation after generation of protein, like a stamp getting pressed into new wax forever. The wax changes, the stamp doesn't.
That's the whole paradox. Solved.
Right. 2. We have to retire this idea that amyloid automatically means disaster. The difference between a plaque that destroys a neuron and a clump that saves your childhood memory isn't whether it clumped, it's how it clumped, and whether a chaperone like Funes was standing there managing the whole build. Structure and control, not presence versus absence.
Precision, not panic.
Exactly. And 3, the one that's going to stick with me longest. This reframes Alzheimer's, schizophrenia, even PTSD as the same basic question. Not how do we nuke the protein, but what broke in the machinery that's supposed to build it, or take it down, correctly. That's a completely different research direction than we had yesterday. But here's the thing. Knowing that your brain runs on this beautifully engineered system of novelty, repetition, meaningful stimulus, timing, that's not the same as actually training your brain to use it well. One's trivia, the other's a skill. That's the gap Noreko is built to close. Spacing things out, forcing retrieval, making the stuff you're learning feel like it matters enough for your brain to bother lighting that match in the first place. If any of this made you want to actually build something permanent up there, come learn with us at noreko.com. Well, that wraps up our episode. If you're still listening, I wanted to thank you for sticking with us through this journey. And we hope you learned something new, as that is our goal with the Noreko Project, to teach people new things and help them remember them. We hope to see you next week again, and we will continue to tackle and dive into even more interesting topics. Have a nice rest of your